These complicated digestive tracts are governed by a whole set of complicated controls, to the point where the stomach not only has its own nervous system, but it has its own system of endocrine hormones to control everything from how food is processed as it moves through the gut to the intimate relationship between appetite, digestion, and cognition. This is a very complex system, which we are far from understanding and still are learning more about every year. We'll try to at least scratch the surface of what we do know here. Processing a meal through the digestive tract involves transferring a meal sequentially through a series of different operations. Control of the digestive tract's function can be divided into roughly 3 sequential phases. The first phase is the so-called cephalic phase, which is involved in various levels of appetite control and preparation of the stomach for the meal to come. This latter level of control is important for efficiency's sake. It doesn't make much sense to keep the stomach prepped and ready to go to receive a meal, that is, to have ample levels of acid in the stomach, all the digestive enzymes in place, and so forth, unless there's a meal actually to come. The cephalic phase helps to activate the stomach when there's a meal actually coming. Appetite control is a deeper phenomenon of cognition, which we'll come back to in a little bit. For now, let's go to the next phase, which is the so-called gastric phase. In this phase, food is subjected in the stomach to acids, digestive enzymes, and so forth, and of course, the digesting meal has to be physically mixed fairly vigorously. This takes some time, and an important aspect of the gastric phase of digestion also involves preparation of the small intestine to get ready to receive the digested meal. The reasons for this are similar to the control of the gastric phase by the cephalic phase. It makes very little sense to have the small intestine ready with the bile salts, bicarbonate, and digestive enzymes unless there's a digested meal actually coming. The next phase is, of course, the intestinal phase. As we've seen, this is where absorption and emulsification of fats takes place, but for the purposes of control, the intestinal phase also feeds back to the stomach, basically to tell it that it has completed its job, and to feed back onto the cephalic centers that control appetite. Again, we'll get into the whole issue of appetite control a little bit later. Let's start with the gastric phase of digestion. The gastric phase is initiated by input from the cephalic phase that basically gets the stomach ready to receive a meal that will be coming. This comes from the brain, primarily through nervous input from the vagus nerve. This nerve innervates several parts of the stomach, including the pylorus, which is responsible for secreting a protein hormone known as gastrin. Gastrin travels through the stomach's circulatory system and activates tissues in the fundus and elsewhere to begin secreting protons, acidifying the stomach, and pepsin to begin getting the stomach ready for protein digestion. Gastric secretion itself is under various levels of control, and we can summarize this with a diagram of inputs and outputs. The principal output of gastrin, that is, the effect that gastrin stimulates, is, as we have said, the secretion of protons and pepsin into the stomach. What stimulates the release of gastrin is, among other things, input from the vagal nerve, which is a top-down control of gastrin secretion from the cephalic phase. In addition to this, there are several local aspects of control of gastrin secretion that are properly part of the gastric phase of digestion. For example, physical stretch of the stomach, as might be expected when an empty stomach has to stretch to accommodate a meal, can by itself stimulate gastrin secretion from the pylorus. Also, the presence of protein in the stomach can itself stimulate the release of gastrin. Again, this makes sense. If there's a lot of protein there will have to be a lot of acid and pepsin secreted. And this comes about through a lot of gastrin secretion. The effect of protein is also, of course, part of the gastric phase of digestion. We turn now to the intestinal phase, which has 2 components to it. The first is to prepare to receive the acid chyme that will be coming in from the stomach. and the second is to feed back onto the stomach to tell it that its job is done. Here's an outline of where the action takes place. The pylorus of the stomach feeds into the duodenum, which is the first part of the small intestine. As we've already seen, at the duodenum, the gallbladder, fed with bile from the liver, and the exocrine pancreas both feed their contents into the duodenum. In humans, the pancreatic duct and hepatic duct join into the common hepatopancreatic duct, but in other vertebrates, these two ducts can be separate. The intestinal phase of digestion begins when chyme is transferred through the pyloric sphincter into the duodenum. There, the presence of protons and fats stimulates the release of a protein hormone from the duodenum known as cholecystokinin, or CCK. That's a pretty big name, but it has a fairly simple meaning. The kinin part simply means to activate, and the cholecyst part simply refers to the gallbladder. So the word cholecystokinin simply means activation of the gallbladder. CCK does more than this, though. When CCK is released from the duodenum, it activates the gallbladder to release bile into the duodenum, where it can begin to emulsify the fats that have just arrived there. CCK also activates the exocrine pancreas to release bicarbonate into the duodenum, so that the protons that have just arrived there can also begin to be neutralized. Both of these stimuli are initiated by the presence of fats and acid in the duodenum. If we look at our box diagram of inputs and outputs, the release of CCK is stimulated by the presence of acid or fatty acids. The response to CCK is the release of bile and bicarbonate into the duodenum. The next set of hormonal controls also involves the duodenum being able to sense the presence of partially or completely digested materials. One of them involves a feedback of the duodenum onto the stomach, that is, of the intestinal phase back onto the gastric phase. There are two sets of these controls. The first is a protein hormone known as secretin. Secretin is released by the duodenum in response to the presence of fats and protons there. The secretin-secreting cells are located downstream a bit in the duodenum, and these serve partially as a fail-safe mechanism just in case cholecystokinin did not do its job completely. If the chyme is still too acid, for example, this can only mean that CCK-stimulated bicarbonate secretion by the exocrine pancreas was not sufficient to completely neutralize the chyme. This is where secretin comes to the rescue. If the chyme is still too acid, secretin will stimulate the exocrine pancreas to release more bicarbonate. The same goes for unemulsified fats. If CCK-stimulated bile release was not sufficient to completely emulsify the fats in a meal, the duodenum will sense this, and secretin will give the gallbladder an extra boost, so to speak, so that more bile can be released into the mix. Let's express this using our box diagram of inputs and outputs. Acid in the duodenum prompts the release of bicarbonate from the exocrine pancreas, and fatty acids in the duodenum stimulates the release of secretin, which stimulates the gallbladder to release bile. The second level of control involves another protein hormone known as gastrointestinal peptide, or GIP. GIP is released from the duodenum by the presence of fatty acids and monosaccharides, such as glucose and fructose, in the duodenum. Gastrointestinal peptide feeds back onto the fundus of the stomach, where it acts to suppress acid production. It also feeds back onto the body of the stomach to inhibit gastric motility. Thus, gastrointestinal peptide acts as a negative feedback of the intestinal phase of digestion onto the gastric phase of digestion. Again, expressing this using a box diagram of inputs and outputs, GIP release is stimulated by monosaccharides and fatty acids and feeds back onto the gastric phase of digestion to inhibit acid production and gastric motility. This system of hormones helps coordinate the complex and specialized processing of foods as they move sequentially through the gut. We've concentrated here on the relationship between the gastric phase and intestinal phases of digestion. The cephalic phase, which we've paid little attention to up to now, is tied up in more complex mechanisms of appetite control. Let's now turn our attention to these.